After doing 117 hand grips, napagod ang arms and hands dahil sa muscle fatigue. According to the review of Constantine Tedioso and Constantine 2021, during each grip, the motor neurons repeatedly send signals to the muscle fibers, causing them to contract. Over time, the fibers use up their energy and accumulate metabolic byproducts, which reduces their ability to contract effectively. This triggers the nervous system to decrease the strength of its signals, a protective mechanism kaya humihina ang grip and the arms and hand feel tired after so many repetitions.
Body temperature significantly affects muscle function, with an optimal range of 37 to 40 degrees Celsius supporting maximal performance. This is evident in a signature produced at normal body temperature, where hand movements and writing appear controlled and steady. In order to visualize temperature effects, some experiments were conducted such as holding ice for 5 minutes straight to stimulate cold exposure, reducing blood flow and dexterity which stiffened muscle and resulted in shaky uneven signatures. Afterwards, pouring warm water in the hands for a few minutes enhanced vascular flexibility and coordination, resulting in smoother handwriting. This finding highlight how temperature outside the optimal range disrupt neuromuscular efficiency, directly altering signature consistency and precision.
Before any tests, we observed the frog's behavior. Under normal conditions, the frog was very active, hopping away from the tray as we tilted it side by side, displaying quick movement and awareness. However, after being single-pithed, the frog stayed still in the tray despite the tilting, because pithing disrupted the main parts of the brain that control movement. Finally, after the second pith, the frog's brain and spinal cord was completely destroyed, resulting to it being paralyzed, laying flat on the tray with no observable voluntary movement. In the normal frog, applying acetic acid triggered a quick and targeted scratching movement. After single pithing, the frog still showed reflex but is much weaker and less coordinated. In a double pithed frog, the scratch reflex was completely not observed, since the brain and spinal cord were both destroyed.
The flexion reaction in the normal frog exhibits a rapid or strong contraction of muscle and is clearly observed when electrical stimulus is applied. While in the single-pitted frog, when the electrical stimulus is applied, it exhibits a slightly weaker response of muscle, but the reflexes are still present. Lastly, in the double-pitted frog, there are still muscle response, but is minimal because both the brain and the spinal cord signals needed for flexion and muscle activation are disrupted.
In a healthy frog, touching the cornea triggers a blink reflex, which is mediated by the frog's brain through its cranial nerves. This action begins with a stimulus to the eye. Its sensory nerves in the cornea then transmits a signal to the brainstem, which sends a motor command to the eyelid muscles to close, thus protecting the eye from damage. After single-pithing the frog, we observe that corneal reflex is still functional, but at slower rates as the frog blinks. This can be attributed to the damages in neural tissue brought about by pithing. Double pithing, on the other hand, destroys both the brain and the spinal cord. As a result, no corneal reflex occurs and the frog no longer blinks when the eye is touched.
So for us humans, this corneal reflex can be observed kapag tayo ay napupuwing or kapag nakakakita tayo ng shiny objects na masakit sa ating mata. So dito tayo napapapikit. So this is to protect our eyes from the damage and to prevent further injuries.
Let's move on to reflex withdrawal. Under normal conditions, the frog was fully active and responsive as we pinched its leg, resulting in a reflex withdrawal. After single pithing, the frog lost voluntary movement since brain was destroyed. After double pithing, the frog showed no response despite any pinching, confirming the loss of spinal reflexes.
Meron tayong kaparehong protective reflex na tinatawag na nociceptive withdrawal reflex. Kapag aksidentally napaso ka o nakahawak ka ng mainit na bagay, for example ay plancha, hindi mo na kailangang mag-isip. Matik, mabilis mo nang aalisin ang kamay mo. Ito ay dahil sa mabilis rin na communication ng ating sensory neurons, spinal cord at motor neurons.
For the first treatment sa dalawang rats, we sprayed acetone. Acetone acts as a chemical irritant kaya na-activate nito ang mga nociceptors. Because of this activation, the rats began showing facial expressions that are part of the rat grimace scale, a reliable tool for quantifying pain through facial pictures. Among the responses we observed were cheek bulging and nose bulging, whisker and ear rising or retracting, and orbital tightening. This reaction appeared immediately after the acetone application.
As we immerse the mouse tail in the 55°C water, the heat activates pain receptors, which send a signal to the spinal cord. The spinal cord then triggers an automatic reflex, causing the tail to flick without involving the brain.
Acetic acid injection in lab rat's abdomen induces acute visceral pain, kung saan nagkakaroon ng abdominal constriction, changes in behaviors, and writing due to peritoneal irritation. Pain peaks within minutes driven by acid-induced inflammation and nociceptor activation.
Lastly, formalin injection is a classic method to test pain reflexes in animals. When formalin is injected into the left toe of the mice, napansin namin ang immediate response ng mice kagaya ng shaking and flicking of the toe and increased flexion reflex.